In this video, you will be guided through the whole test procedure for RIDA QUICK Gluten Quant. The RIDA QUICK Gluten Quant enables a safe, fast and simple quantitative analysis of gluten residues from wheat, rye and barley on surfaces, in CIP water and in foods in test strip format. In this video, we present the contents of the test kit, explain how to prepare different sample types and guide you through the testing procedure. The label on the test kit contains all relevant information regarding batch, shelf life and storage temperature. The printed QR code links you to the EIFU portal, where all kit-specific documents, such as instructions for use, are available. The kit includes a certificate of analysis containing all relevant quality information, as well as a QR code that provides batch-specific data for evaluation using the RIDA Smart App Allergen. Further components are swabs, disposable pipettes, squeezable test tubes, conjugate 1 and 2, as well as swabbing buffer, each provided in a dropper bottle, test strips and extraction tablets. The kit contains sufficient materials for 15 determinations. In the following, we will focus on the analysis of swab samples, CIP water and food samples. Before each analysis, the samples must be prepared accordingly. As a first step, add 4 drops of conjugate 1 and conjugate 2 respectively into a test tube and mix thoroughly. For example, by using a vortexer. For surface sampling, moisten a swab with 8 drops of swabbing buffer. Further, wipe the sampling area thoroughly in a crosshatch technique. After sampling, immerse the swab in the mixture of conjugate 1 and 2 in the test tube and rotate several times in the liquid. Additionally, squeeze the swab repeatedly in the liquid by squeezing the test tube. This process should take approximately 15 seconds. When removing the swab, squeeze the test tube again to extract the liquid from the swab and collect it in the tube. Subsequently, the sample is incubated for 5 minutes. For sample preparation of CIP water, 4 drops of each conjugate are thoroughly mixed as previously shown. Now use a disposable pipette to draw a sample of the CIP water and add 1 drop to the conjugate mixture. To do this, you should draw a significantly larger volume. 1 drop corresponds to approximately 20 microliters. Alternatively, you can use a calibrated piston pipette. Mix the sample thoroughly and incubate the sample subsequently for 5 minutes. For the shown sample preparation of food samples, a rice flour is used. In food samples, gluten is processed within a matrix. Therefore, gluten needs to be extracted prior to analysis. For this, weigh out 1 gram of homogenized sample. Alternatively, use 1 milliliter of a liquid sample. Afterwards, add 1 extraction tablet and 10 milliliters of 60% ethanol to the sample. Further, incubate the sample for 2 minutes and subsequently mix thoroughly for 20 seconds. Repeat this step. Incubate for 2 minutes, mix for 20 seconds. To obtain a particle-free supernatant, a filtration step is required. The filtration step separates insoluble components of the sample and the extraction tablet. For this, filter the sample by using, for example, a fluted filter with a pore size of 9 to 12 micrometers. Collect the flow-through in a new tube. Only a few microliters of the particle-free supernatant are needed for further analysis. Thus, complete filtration is not necessary. For the following analysis, mix conjugates 1 and 2 as previously shown. Further, draw the particle-free supernatant using a disposable pipette and add 1 drop of the conjugate mixture. Mix the sample thoroughly and incubate the sample subsequently for 5 minutes. For sample analysis, take 1 test strip from the tube. The test strips are sensitive to moisture, therefore close the tube immediately after removal. Place the test strip into the test tube. It should rest on the bottom and must not come into contact with the front side of the tube. This is followed by a 5-minute incubation. Remove the test strip after incubation. Evaluate the test strip within 30 seconds of removal by using the RIDA Smart App Allergen. A visual evaluation of the test strip is not possible. Further information on evaluation using the RIDA Smart App Allergen is available in the instructions for use and in a separate video. The RIDA QUICK Gluten Quant enables a fast and simple quantitative analysis of gluten contaminations all along the food production chain. It is the only quantitative LFD on the market using the R5 antibody. The test results are expressed directly in microgram per kilogram gluten, so there is no conversion factor needed anymore. Additionally, processed matrices, as well as wheat, rye and barley are used during the validation process, which is in line with the new AOC guidelines. Finally, the evaluation of the test is performed with the innovative RIDA Smart App Allergen.